So, good morning. My name is Anna Mitchell. I am a consultant genetic counsellor working in the UK, and I've been a genetic counsellor for over 15 years now. I started off specialising in inherited heart conditions, which is the background that I had before I became a genetic counsellor. I have a PhD in congenital heart disease genetics. And following my work in inherited heart conditions, I then went to a company offering direct to consumer genetic testing, and they had a exome product, a clinical exome product, which they needed help, support with genetic counselling for clients who had received results from that test. I'm currently working back in the NHS, and at the moment I'm specialising in inherited cancers. My previous role, I was head of clinical governance for the private company that was offering the genetic testing I mentioned. And during the pandemic, we also built our own lab and got it accredited. So I was director of that lab during the period of accreditation to the international standard 15189, which I'm going to talk a bit more about later in this presentation. So thank you very much for joining me this morning. My talk is entitled Getting Quality Genetic Testing for Your Clients. And I'm presenting this to the Genomics Hub community. Hopefully a few more of us will join this presentation later on. In the talk this morning, I am going to discuss the different types of genetic test available on the market. This is not aimed at being a comprehensive list, just an overview. And then I'm going to move on to understanding the appropriateness of the genetic test that you might want to order and the quality of that test. I'm going to cover a little bit about variant interpretation, and then I'm going to speak about lab credentials and accreditation and quality and what that means in that context. This is all leading towards understanding what best practice in the industry might look like, and we're going to think about a couple of testing pitfalls that you may come up against, and hopefully it'll prevent you from coming up against them. So thinking about the different types of genetic tests, broadly speaking, these are the sorts of tests that I think about on a daily basis. There are things like SNP genotyping, where predominantly commercial companies will offer testing for known single nucleotide polymorphisms that are associated with either disease risk or certain lifestyle factors. I've given an example of 23andMe, but they're not the only people offering this sort of test. But they are probably one of the best known globally. There's targeted mutation testing, where very specific known mutations in a disease gene or set of genes are tested for a particular disease type. The example you can see here is testing maybe 4 different known mutations associated with causing cystic fibrosis. They're mutations in the CFTR gene. Then you might think about a targeted gene panel. So again, the genes have been selected. They're known to be highly associated with a particular disease modality, in this case, inherited ovarian cancer risk. And so there's a small number of genes, depending on the condition, anything from 1 or 2 to 18, 20, maybe more. But the point is that you're still keeping a quite tight focus on the disease that you're looking at. Then there is exome sequencing. Again, I've given an example of circle DNA, which is marketed by a company called Prenetics. They're not the only people offering this out there. So they offer what you would probably regard as a clinical exome. It covers 500 different reports, anything from wellness through to cancer risk. So they have quite large numbers of genes that they look at in each of those disease areas, but it's still constrained to known disease areas when it comes to reporting. And then there are companies that claim to be doing genome sequencing. Dante Labs is one of those. And to be honest, I haven't had enough time to look into whether they really mean that they're offering a genome sequence or whether this is kind of equivalent to the clinical exome that I just mentioned. But questions to ask yourself when you're thinking about ordering a genetic test are, and you've got a lab in mind, what technology is the lab using? Is that the right technology for the gene and the condition? So is it going to carry out the right test, and is the interpretation going to be adequate for you for the specific needs that you have? This may vary depending on what test you're offering and in what circumstance and to which sort of client. If the test is a sequencing test, do you have an understanding of what the read depth is? How reliable is that result going to be, and is it going to pick up any copy number variants? Who is doing the interpreting of your results? Is it somebody who has specific qualifications, and is it somebody who is regulated in any way so that there is a level of accountability and responsibility behind that? What framework does the results interpretation occur under? I'm going to talk about the sorts of framework we might consider, but is there a rigorous approach to how interpretation is done, and is it reliable and repeatable? And is specialist interpretation needed for your particular gene of interest? I apologise, I should have been clicking through, but it was just some illustrations. The one I wanted to particularly talk about, which is a good example of picking the right technology for your test, is if you have a client who's concerned about alpha thalassemia risk. Alpha thalassemia can, of course, be caused by both single nucleotide mutations and deletions. So you need to make sure that the test covers all of those eventualities. Otherwise, you're only going to get part of the picture of the risk for that client. When I talked about frameworks, this is one of the possible frameworks, and we'll come on to that in a second. So this was a paper that was published back in 2015 by Richards et al. They published these guidelines with the aim of serving as a framework for variant classification. And they did a reasonably good job. It has subsequently been adopted worldwide by many labs. And in the intervening time, a lot of the evidence has been reviewed, evaluated, and adapted. But it looks quite complex, but actually it's a really nice, easy read once you've understood what you're looking at. This table gives you sets of evidence that you can use when you are classifying a particular variant that you might have found in a patient. And it helps you evaluate, it helps you weight that evidence. So, for example, up in the top left-hand corner, we can see strong evidence that a variant is benign. Might be that the minor allele frequency is too high in the general population. So basically it's saying that this variant is something that's just a variation of normal. It's very unlikely to be associated with disease. Whereas if we look over at the top right-hand side of the diagram, in the second row down, there's some very strong pathogenic evidence there that the variant results in a null variation of the gene, so it results in loss of function of the gene, and that is a known disease mechanism for this particular disease. Since 2015, lots of people have been working on refining these variant interpretation guidelines. And what has happened is that many expert panels have been set up to really curate the known variants in a lot of these genes and provide really strong evidence for pathogenicity or otherwise. Of course, this takes a great deal of time and effort, so a lot of these are incomplete and they don't cover all disease areas yet. But some examples include one panel that has focused specifically on the cardiomyopathy gene MYH7, which is a myosin heavy chain gene. There are 18 other genes that can be associated with cardiomyopathy, well, and more, depending on what flavour of cardiomyopathy you're looking at. So you can see that there's a lot of work still to do in that disease area alone. The Marfan gene, fibrillin 1, there's a specialist panel for that, and also for the cancer genes ATM and PALB2. So this is an example of how that variant interpretation framework has been applied to thinking about a particular variant. And I've chosen one of the most common variants in the MYH7 gene, which is the cardiomyopathy gene I just mentioned. But you can see down at the bottom in the comment, the evidence supporting the pathogenicity of this variant is clearly set out. And also it's really nicely signposted. So after each piece of evidence, the point in that table that we saw is referred to so that you can see exactly which piece of evidence has been used. It's important to know that you should not use any piece of evidence to meet more than one of the guideline criteria. So basically you can't use something twice when you're making this assessment. So over that time, it has been acknowledged that not all pieces of evidence should be applied for every genetic disease. So allele frequency can sometimes be misleading, by which I mean the prevalence in the general population. Some genetic conditions are genuinely pretty common, which can muddy the waters a bit. And also that does depend on the population that you're looking at, of course. In addition, some categories of evidence have been withdrawn altogether. So there are two mentioned there which relate to other databases. If you're interested to know what they are, you can go and look at the table. And I've given some examples of those expert working groups that I mentioned earlier, who are refining the frameworks for individual genes. And the QR code will take you to that website that I've listed at the bottom. This has all kind of led to many people trying to write a more definitive, refined version of the guidelines, and this is one example of that. So in the UK, the Association for Clinical Genomics Science published this paper in 2020, which is their suggestions for how to refine the interpretation framework, making it a bit more practically applicable in real life, and also acknowledging the areas that maybe didn't work so well when people tried to apply Richard's initial framework. The other thing to consider is who might be interpreting the results in the lab when they're going to write the report for you. So is it somebody who is qualified as a clinical scientist? Is it someone who's qualified as a genetic counsellor, or is it a researcher? And this is important because clinical scientists are often regulated. Certainly in the UK, they are mandatory regulated now, and there's actually a move for genetic counsellors to be regulated to a similar level and regarded at a similar level. So actually, when we become mandatory regulated, we will be under clinical scientists as a category in that body. In the US, there is a system of state licensure. So each state has its own requirements, and not all states have signed up to requiring licensure from clinical scientists. It does also mean that if you move states, you have to gain new licensure in the state that you want to operate in. And in many other countries, there's a system of voluntary certification whereby practitioners will sign up voluntarily to follow a code of practice and be publicly signposted as reliable people, but there's no formal regulation, which basically means that they're great systems, but it means that somebody can't be stopped from practicing if they are not following the guidelines, or even worse, practicing dangerously. A similar sort of system applies for genetic counsellors. So there's state licensure in the US, there are voluntary registration systems in other countries, including Australia and the UK. And as I said, the UK is moving more towards a mandatory model, but that's not yet in place yet. And I understand that in UAE there is licensing for genetic counsellors, and I haven't done a lot of research into other countries. I'm sorry about that. Researchers interpreting results may well be subject matter experts, but sometimes it depends on their background. They may not have the knowledge of clinical applications, so you may need the input from a clinician to help with that interpretation. And oftentimes, I think this is less applicable now. I think people understand more about the need for consistency and rigor and repeatability in interpretations. But certainly in the past, there used to be a great deal of variation and less of that rigor. So I've talked about lab accreditation, but I haven't actually discussed what it is yet. A lab can be accredited or certified to different levels or standards. So it's really important as practitioners to understand what these levels are and how they relate to one another. Accreditation means that the lab meets a certain standard, and they should be published in the public domain so that you can check them in a transparent way. There are international standards that are published that relate to labs and specifically to genetic testing. Those standards are ISO 15189 and 17025, and I'll talk a little bit more about them in detail in a second. Different countries also have local standards. So in the USA, there is a system called CLIA. So that is the Clinical Laboratory Improvement Amendments. That's a certification program. And in Saudi Arabia, the Central Board for Accreditation of Healthcare Institutions has published national standards for medical labs and blood banks. Again, these are just examples. They're not intended to be a comprehensive list. Not all standards are created equal. You need to know what they are standards for before you can evaluate whether they're helpful and appropriate. So the one that is the gold standard internationally for medical laboratories, and I would say for any genetic testing laboratory, is ISO 15189. It has recently been updated following the pandemic, and it sets out requirements for quality and competence in laboratories carrying out testing for medical purposes. It is based on the other international standard that I've put up on this slide, 17025, and the basis of ISO 17025 is setting out general requirements for the competence of testing and calibration of laboratories. The focus of 17025 is a lot on what they term metrological traceability, so high standards of repeatability in the way that you measure things. Of course, with genetic testing and medical testing in general, often there's a layer of interpretation that needs to go on top of those numbers, and ISO 15189 also incorporates that need as well. So the International Organization of Standardization is the body that creates these standards. It's a non-governmental organization, and it's designed to bring together expertise and to share knowledge. It's voluntary, it's consensus-based, and they're designed to meet a need globally. I found a really helpful paper which talks about accreditation in the EU countries, which I've linked on this slide. Accreditation is mandatory in some European labs, And there was a helpful quote which says it's not about who the best is, but who has a system of standard procedures which aim to improve the quality and patient safety when it comes to results. There are lots of different bodies across the world that can carry out accreditation. In the USA, there are two that can accredit to ISO 15189, and they are called CAP and A2LA. CAP is probably the best known, and a lot of labs you will see in America are what they call CAP Clear certified. So CLEAR is the standard I mentioned earlier, which is a certification minimum standard in the United States. That's defined by the FDA, and it focuses on technical procedures. It's peer assessed, and it is not the same as standard as 15189, which is why CAP have introduced an accreditation program to get labs up to 15189 level. There are many fewer labs in the USA who currently hold that standard because that focuses on quality management and it is professionally assessed, and it's a much bigger undertaking than gaining CLEAR certification. In Australia, there's an organisation called NATA. In the UK, there's an organisation called UCAS, which is the UK Accreditation Service. And I mentioned the Saudi Arabian body earlier, which is the Central Board for Accreditation of Healthcare Institutions. Many of these organisations, I've definitely seen this for NATA and for UCAS, will provide accreditation for labs in other countries, especially where there is no local framework for accreditation. So that's helpful to know as well. If you see an accredited lab in, let's just say, Indonesia that has been accredited by NATA, that is a real thing. First of all, I would have thought, oh, is that really right? But yes, apparently it can be. So this standard, as I've mentioned, means that the lab operates and monitors its operations in such a way that the quality of their results can be assured because they are reproducible, transparent and reliable, as well as following clear guidance and meeting external quality assurance metrics. So a lot of labs sign up to quality assurance programs where they will receive samples from a standardization body, which they then report to that standardization body to show that their tests work in the way that they are supposed to. So one of the helpful things I saw on the College of American Pathologists website, the CAP website, was a testimonial about a lab that had moved from that CLEAR certification to ISO 15189. And they said that before they did that process, they thought they were doing quality well, but they realised when going through the process that what they were just doing was remedial action, which means if something went wrong, they were fixing it. What they weren't doing was putting procedures in place to make sure that it didn't happen again. And the way you do that is through root cause analysis and things called both corrective actions and preventative actions. And they said that following the framework of the ISO 15189 standard got them to that level. And they realised that they were much more competent and providing much better quality service as a result. So why is quality important? I think we all understand what quality is day to day, but quality when we're talking about regulated procedures or highly technical procedures is really important when we're thinking about patient safety. We need to be assured that those results are reliable and that we're not going to give the wrong information. We need to understand the accuracy levels of the test, so how certain we can be when we're giving people information. And we need, ideally, for labs to be able to update us if the interpretation of the results changes over time. And of course, with genetics, that happens all the time. So we do need to be kept up to date. So you might be saying, well, this is all great, Anna, but there's a lot of information here, and I want to know how I can apply this in a practical way. Well, the first thing you need to remember is that the lab should hold accreditation for the specific test that you're interested in if you want to be really sure and reassured about those results. As an example, if the lab holds accreditation for testing levels of cholesterol in a blood sample, that's brilliant. But you want to submit a blood sample to test the cystic fibrosis gene for mutations. It doesn't hold accreditation for that test. Then you would probably be less reassured about the likely quality of the CFTR test compared to the cholesterol result. What may happen is if you ring up the lab, they may say, oh, well, actually, we're working towards accreditation for the CFTR test as well, which does provide you with a level of reassurance, but not as much reassurance as if they've actually been granted that accreditation, of course. As I mentioned before, accreditation should be published and in the public domain so that you can check them. And I've given the URL here to my next slide, which is an example of one of the local labs that I work with, which has been accredited with the United Kingdom Accreditation Service, that UCAS body that I pointed to earlier. You can see this is the top of their schedule. It's many pages long, but this is publicly available on UCAS's website. There are clear contact details for who runs the lab and how you could contact them. And then the remaining pages provide a detailed list of all the input tissue types that the lab can accept, all the test types it can carry out, and all the analysis and interpretation frameworks that it uses that have been evaluated by UCAS. They probably also carry out procedures that have not been evaluated, but they can't be published on this list. So when I'm looking at a lab report, I want to see clear and transparent detail about exactly which part of which genes have been tested, which test technology was used, and if a variant has been found, detail about how the interpretation has been carried out, how the conclusion was reached, and ideally, which pieces of evidence have been used from those ACMG guidelines in 2015 to create that evaluation and assessment. These details help me to evaluate the evidence for myself and then provide clear information to my clients or patients about their risks. And that it means that I can be assured that that evaluation is based on whether the appropriate bits of the gene have been tested, whether the right sort of test was used, and how reliable those results might be. I've got some examples that we can talk through for the remainder of the session, and then I'd be delighted to have some questions. The first example looks at the sort of testing that 23andMe offer. I've taken cystic fibrosis as an example here, and you can see on this example test report that the delta F508 mutation has been identified in this person. The package insert provides detail on the sorts of technology that was used. It was an Illumina bead chip, which is an appropriate test when you're doing assessments of single nucleotide polymorphisms, which is what 23andMe largely do at the moment. It's not the only thing they do, but it is still the bulk of their testing. So we can see that the test technology, it's not defined on the report, but it is clearly linked in the report how you find that. And the company have tested single nucleotide polymorphisms commonly associated with causing cystic fibrosis. And the interpretation framework is based on the fact that only cystic fibrosis causing SNPs have been assessed. So in a bit more detail, we can see that the caveats around this test are clearly displayed. That it's clear that they've only tested 29 known variants. They've also made it clear which ethnicities this test is appropriate for and provided information about each of those ethnicities in terms of the reliability. So you can see that it is variable in that across those different ethnicities. It is clearly set out that the test does not cover all variants that cause cystic fibrosis. and the package insert provides information about accuracy and reproducibility. This all makes sense to a healthcare professional, but it may make less sense to a layperson or customer, who is typically the sort of person purchasing this test directly. And there's no clear indication of the person who was responsible for the report, probably because it was interpreted by an automatic algorithm. So 23andMe do these things well. They are very transparent about everything, which I like. The information is there to be found. They're not trying to hide it away. But as we can understand, there are certain limitations to the test which have been declared. So this test offered by 23andMe does only test the most common variants. In many labs, that is still the standard initial test, but a lot of labs will also move on to sequencing if they don't find anything when they think they should be able to find something. So a lab generating full sequence data will be able to detect and evaluate any sequence variants which are detected anywhere in the gene, rather than just focusing on those 29. And therefore, to an extent, a negative result from this 23andMe test is going to be less informative than a full sequence. And I think it's just important to think about that and evaluate it when you are thinking about what the patient in front of you wants and needs to know in terms of their risks. This second example was generated by a company called Gene Health, who use a lab called Health Encode, and it is a BRCA2 result. So we can see that both sequence variants and copy number variants in BRCA1 and 2 have been assessed with this test. A variant has been identified, and the interpretation of it being likely pathogenic the report says they've applied the American College guidelines, which are those ACMG guidelines published in 2015. They haven't set out how they have applied them, but at least they should have some ability to tell you how they've applied them if you wanted to know. And it's very clear that real human beings have been responsible for checking this report and signing off on it. Then on further down the report, we can also see that the method of sequencing is clear. There's a read depth of about 20x, which is fine. It's okay. Now most labs do better than that as a minimum, and it can be helpful for assessing dosage, so copy number variation, as well as single mutations or insertions and deletions. So it can give you a steer as to the reliability of the result. And there is also published accountability about the reporting framework and the responsibilities that they regard as theirs for re-examining results. So if a result was uncertain, there is an undertaking to review it in a certain period of time and to pass on those findings. So this particular report meets all of the criteria that I would like to see in a report. And similarly, this report from an NHS lab in the UK does. A clear variant has been found. It's clear how many genes have been tested and coverage. We've got a read depth of 50x and all genes are covered 100%. So that's helpful. We can also see that an interpretation has been set out very clearly. It is a bit light on detail, but it tells us that the variant hasn't been seen in the literature before. Similar types of copy number variation are known to increase cancer risk when found in this gene, PALB2. And the conclusion leads us to understand that the lab have a level of uncertainty about this variant, but they believe that it is associated with increasing cancer risk. We can also see at the bottom of the report that there are real people who have reviewed it and signed off on it. They are registered clinical scientists, and we've understood how they've reached these conclusions. On the back of the report, we can also see that it is fit for purpose. It detects single nucleotide variants, indels, and copy number variants. We're clear on which test technology has been used, and we're also clear on that read depth of 50 times, with the regions of interest being covered to 100%. And finally, I thought I would just do a comparison between the gene health BRCA2 variant that I discussed in example 2, and I looked up the same variant on ClinGen. Just like we saw in that cardiac example earlier from ClinGen, some labs are really good at signposting how they have arrived at their interpretation, listing each piece of evidence. So you can see that I've highlighted there each of those pieces of evidence that is clearly set out in this interpretation. So the cancer variant interpretation group do agree with the interpretation that the variant is either likely pathogenic or pathogenic, and they've set out their reasons for classing it as pathogenic. So I hope that in this presentation I have set out some helpful pointers on how you might choose the right genetic test for your patient, and how you might evaluate the quality and appropriateness of that test. I've discussed variant interpretation and that there is a framework and a level of rigour that can be applied to it, which I hope everybody was aware of, but it's often worth thinking about these things. I hope I've also set out what quality is when we're thinking about labs and how reliable and how competent people are. Basically, a lot of labs will be very reliable and competent, but this gives you the assurance that somebody else has checked that and that they meet a certain standard, and what best practice might be. And hopefully this will help those of us who do see patients to provide quality and safe genetic counselling for our patients in the future. Thank you very much.